Great. As I figure out, there we go. Yeah, so I'm Sam. About 13 months ago, I started as I started my lab at the Crick Institute in London, which is where I guess all of you guys are as well. I had a little bit of a non-traditional path to get to where I am now, which is maybe something that you guys will be interested to hear about. So I started out actually as like a theoretical physicist. So in fact, if you like go and you look, you like dig back in my in my papers, you'll find this like paper from 2013 or something on like measuring entanglement in like systems of many qubits. So it started out like really doing like quantum information theory kind of hardcore. And that was split between Harvard College, which is this small liberal arts college in the US. We have these, in the US, we have these quote unquote liberal arts colleges, which are basically have no graduate students. They only have undergrads. And I went there because these colleges still do some research, but when they do research, because there are no graduate students, it means that it's all being done by the undergrads. So like it's a place that you go if you really want to do like undergraduate research. But you know, they don't have very good like international profiles. So I then did a junior year abroad at the University of Cambridge, which was the first time that I was actually like living in the UK. Absolutely loved it. It was a phenomenal place to be a student. Now I go back and Cambridge just feels very small, like compared to London. But, and again, that was all basically doing theoretical physics. But afterwards, after I finished my undergrad, I ended up feeling like there were really like no unsolved problems left in physics, which is maybe a little bit of an exaggeration, but you know, as a scientist, you kind of always want to be in whatever the area is where like the most exciting things are happening, right? And I definitely felt at that point like the most exciting things in physics, you know, were at this point like 40, 50, 60, 70 years old, right? And I wanted to be somewhere where new things were happening, which at that point already in 2013 or so, it was pretty obvious that that was in you know, biology and biotechnology and synthetic biology, where, you know, if everything is solved in physics, then like in biology and neuroscience, basically nothing is solved. And so I went to MIT for my PhD and kind of quickly realized that the difficulty in biology and neuroscience is not so much a lack of you know, theories, which is what which is what I had been doing in physics, but rather a lack of data and a lack of the tools that we need in order to get the data. And so I spent this. So I did my PhD at MIT, basically inventing new tools for biology and neuroscience. And you know, I'm really an inventor at heart, and I'm an engineer, and I invented a bunch of different things. And so three of the kind of biggest, the big ticket ones are just on this slide. So the top one is SlideSeq, which is a new way of measuring the locations of different cell types and different RNA in tissue. It's like an omics method. It's a spatial transcriptomics method. So asking not just what kinds of cells are in the tissue, but also where those cells are located. Then below that in the middle is timestamps, which was like the first temporal transcriptomic method. It only worked for a single RNA. We're now working on, but basically what it allows you to do is actually look back in time and say not just what transcripts were in the cell, but also when were those transcripts created, so you can figure out, you know, when the cell was doing different things in its history. And the whole idea here is kind of like contextual omics. Like, you know, we want to ask not just what are the cell types, but where are they? What are they doing? You know, what is their history? How did they get to where they are, right? And actually maybe that one is, yeah, maybe I will tell you guys the story about timestamps as well. It's like a little bit of an interesting thing how we got there. But the third one, the third technology is implosion fabrication, which is down here at the bottom. which is a completely like unrelated, is a nano-fabrication technology where you can actually like embed materials inside of a hydrogel and then shrink it down to the nanoscale. But so this is all just to say I worked on a bunch of different things at MIT and a bunch of different like very applied technologies, and got very frustrated with the difficulty of spinning those technologies out, right? So it was stuff like that even at MIT, which is held up, like especially over here, is held up as like the paragon for, translation, for technology translation. Things would happen like with implosion fabrication, that we were about to submit the preprint one day, and I was in my second year of grad school, and I had to stop and say, wait a minute, guys, like, do we need to patent this before we submit the preprint? And the answer to which is yes, you do. Yeah, you definitely do. And like, this was in one of the labs that is thought of as like one of the most entrepreneurial labs at MIT, which is one of the most entrepreneurial places. And still, it's like me, the second year graduate student, who has to sit there and say, wait a minute, do we need to patent this? Right. And so I just was like, this is not like, academia is systematically bad at, doing this kind of translational, like figuring out how not just to like write a paper, but also how to get your technology out into the world. And I wanted to figure out how to do that better. Right. So afterwards, I didn't do a postdoc. I instead did an entrepreneurship in residence at Petrie, which is a biotech incubator in Boston, and worked with them on starting some companies around different technologies that I had invented. And so we started 3 companies, or I co-founded 3 companies over the course of that year. One of which was Helios, which was developing a drug delivery device for the gastrointestinal tract, one of which is still in stealth, and one of which is Saturn V, which is a really kind of crazy project that is building like a robotic system to drill microscopic holes in the head for like futuristic brain-computer interfaces and for drug delivery. Like literally like 300 micron holes all the way through the skull, so that in like 10 seconds under local anesthesia, so that it just like makes access to the brain like super, super easy. So spun out those companies, and then, and actually at this point, there are 2 of the other technologies I invented in grad school are also being commercialized. And so I've gotten a lot of experience with commercialization, and then came here to the Crick, where, I founded this lab, the Applied Biotech Lab, where the goal is to kind of combine the experience of like basic foundational technology development from MIT with this like experience of company creation and translation from Petrie, to figure out how not just to build technologies that are going to change the world, but also how to get them out there so that they actually can change the world, right? And so we do this across 4 different areas. Oh, this is a little bit outdated, but across 4 different areas. We have some work on like liquid biopsy technology, so figuring out how to diagnose diseases 10 or 15 years before onset. We do a bunch of work on viral vectors for gene therapy, so figuring out how to deliver viruses in a programmable way to specific cell types throughout the body. We do some work on and then we do some work on omics, basically, which is what's here on the right. So like we do some spatial transcriptomics work, but we also do some connectomics work, which is to say figuring out how to map connections between neurons in the brain at like single synapse resolution. So just like a lot of different projects, all kind of unified by the fact that they would have, like, if they're successful, they would have a major impact on science and on medicine. Now this slide. So I just pulled up like this old slide deck, and so now this is like getting really old. So this is like kind of where we are right now. So back then we had 7 people. We now have something like 10. And some of the ways in which the lab is organized are like are a little bit different from a standard academic lab. Is that keeping with the goal of trying to build a lab that can like facilitate not just the invention but also the translation of these technologies. We have an advisory board, which are like people who are outside of academia. Well, except for George. George is on it, but he's an academic. But the rest of them are like successful entrepreneurs or successful investors people who have built like giant companies, who have run giant companies, who have founded a lot of companies, who we can like go to, you know, just for advice about like, we're working on a technology. What do we, you know, what is the best thing to do with it? Is this a technology that can actually get out into the world and have impact? And if so, like, what is the right way to do it? We also maintain like a network of many VCs, investors, because basically like the thing about investors is that their goal is to kind of know what the unmet needs are, or like one of their jobs is to know what the unmet needs are in the market. And so, you know, by maintaining friends with them, you get to hear about what are like the trends, and also what is like currently investable, and what is and like what are the real needs. Like, for example, like what are the drug trials that are failing, and why do we think that they're failing? So we maintain a really strong network of those. And we've got, we're extremely well funded. Again, this is super outdated. I've gotten a lot more funding than that now, but basically, yeah, we have an extraordinary amount of money. So if anyone wants a job, we have a lot of positions available for jobs because we're really well funded. Okay, let me switch over to a different presentation. And also take this opportunity to ask if anyone, like I said, it's like super informal. So does anyone have anything they want to ask me about my career?
Go ahead, AG.
Yeah, I wanted to ask, like, you spend like about more than 10 years in your biosciences. How did you find it, like, after, you know, joining in MIT, creating your own, you know, patented technologies, and, you know, now you're thinking of businesses, like, how did you feel as a journey from there to where you are now at this moment?
Oh, I mean, how does it feel? It feels awesome. Super exciting. I mean, the thing, so basically like this thing about commercialization, right, is that why do we commercialize? Like, there are a couple of different reasons that you might want to commercialize things, right? Obviously, one reason that you might want to commercialize things is that you want to get rich, and that's great. And I think that you know, the work that people do in academia and like this kind of technology work is like super important. And you know, if you can get rich off of if you invent some technology and that technology goes out and changes the world, you definitely should get rich off of that. So that's great. But like, that's like not really the goal, right? I mean, the goal is that the reason that we commercialize stuff is because it's a way of leveraging capital for science, right? So companies, like when you're in academia, you can get this impression that like that all of science happens in academia, and that's like cannot be further from the truth. Like, the companies do fantastic research. Like, biotechs and pharma companies do fantastic research. And if you build something in a way that allows it to be commercialized, it allows you to leverage like 10 or like 20 or 30x more funding for that thing than you can possibly leverage in academia, right? So like this project timestamps that we're working on, if we develop it in a way that can be commercialized, then like rather than doing it on like a $1 million grant with a postdoc and a graduate student, right? All of a sudden it starts out that way, but then it gets turned into a company with $100 million worth of funding and like, you know, whatever, 50 people on staff working on it, right? And that's just like an opportunity to do like so much more exciting like science at scale than you can possibly do in a lab, right? And this has also been like a little bit of a theme throughout my career, because one of the things that if you go and you look me up, one of the things you'll find are these focused research organizations, which is an idea that I came up with for like a new way to fund science at scale. And a lot of what I do is think about like, how do we actually scale it up? Like, how do we leverage more capital to do more science, right? Leverage more capital and leverage more talent to do more science. So I don't know if that answered your question at all, but.
No, it answers it pretty neatly. Actually, like after graduating and all, I was also looking for options to, you know, not now, but like in 5-10 years, starting a company and, you know,
Yeah.
build it up on that. So having someone who has already been through that, and it feels awesome at the end of the day.
Oh yeah.
Feels really nice to, you know, think about like, you know, you want to start those things as soon as possible, I would say.
Yeah, for sure. Any other questions?
I would just like to ask, since you've had a wide experience in different fields of science and then multiple projects under your belt, how do you find managing all of that and balancing everything that you're doing?
Yeah, it's hard. I mean, I think that it's tricky to figure out how to do it. Because like the canonical thing that you do as a scientist is that you focus on like one thing and you do it, right? And what we do is a little bit different, which is that we have like specific areas of expertise, or like we have tools that we know how to use, and we like figure out what we can do with those tools, right? And it's hard because you're always working in an area where you're not an expert, which is something that like your PhD like committee will tell you is an awful idea. And I actually don't think it's an awful idea, but like the PhD committees, especially here, they really hate it, like because they're very risk averse, right? But the thing, but like you figure out ways to manage the risk, right? So there are a few things. Like the first thing is like fail fast, and this is just like this should always be your like the motto, right? Which is figure out really quickly if your idea is going to work. Or sorry, if your idea is not going to work. So whatever you think is most likely to make your idea not work, go and do that first, right? So in our case, what that means is like we have some idea, and because we're always working in areas where like we're always doing things we haven't done before, like half the time we have an idea and we go and we look in the literature and it's been done, and that's great. Like that's fantastic, right? Because that is, you just did a Google search and in 10 minutes you discovered that you don't need to spend 3 years working on something, right? Which is good. But like, so that's like obviously like the first level. Second level is like, go talk to someone, right? You go find if you have some idea about immunology, you go find an immunologist, you talk to them and they're like, yeah, this is stupid. It was done 60 years ago. Great, right? And if eventually you cycle through enough ideas and you come across one where people are like, oh shit, like you might be able to do that. Like, wow, like that could be cool, right? And the point is that if you have 100 ideas, you only need one of them to be good, right? Like no one will remember, no one cares about the ideas that you don't have, or like the ideas you don't have, or like the ideas that don't work. So I think that's basically the thing. Like I list all these projects that I did in grad school, and like the truth behind that is that I started 26 projects in my PhD, and 5 of them worked, and that's great. And I talk about the 5 that worked. Right. But like there were 26 that somewhere I spent like 9 months working on it. And then was just like, this is nonsense. It's never going to work. And somewhere it's like, we had the idea, we like ordered some stuff, we did one experiment and gave up. Right. But that's how you do it. You make sure to fail fast. And you also collaborate. That's like the other way that you do a lot of things at the same time, is that you just have partners, right? You build a team.
You say that you predominantly focus on novel ideas rather than building on other ideas that previously already exist?
Everything builds on other ideas that already exist, right? But like, what I mean is that we usually work on things that we have not worked on before. So we are the best at working on things that we have not done before. Or that's not exactly the right way to say it. It's just that one of the things that we are particularly good at is working in new areas, right? Where like we could also go and work on stuff that we've worked on before, but like lots of people are capable of doing that. And what we're particularly good at is figuring out how to get projects to work in areas that we haven't worked on before. So, but I think that's the other main thing, which is just like, you have to figure out what your strengths are. And this is like, and this is just like part of life, right? But, you figure out what you have that you can leverage uniquely, and then you do that. You kind of milk that. So let me tell you guys quickly about timestamps. I think this is an interesting example, mostly because of the way that it happened. So I'll kind of try to tell you guys. I'll try to give you an example of what one of these projects looks like. So this is the paper. It came out in October of 2020, so a bit more than a year ago. The basic concept, I don't want to spend too much time on the science, actually. You can read the paper. But it was a collaboration between Fei Chen and Ed Boyden, which were 2 of my mentors in grad school. And this is Linlin Chen in the bottom left, no relation to Fei, but she was an undergrad when she started working with me. And she's amazing. She's now doing her PhD at Caltech, and she got a first author Nature Biotech paper. So that went very well. And the basic concept is, how do we figure out what was happening in the past? If we're doing if we're doing a sequencing experiment now, how do we figure out what the cells were doing previously, right? And the fundamental concept, the only thing that you need to know to understand this project, is that asking what happened in the past, asking when an RNA was created. So in order to ask what the cells were doing in the past, we're asking when were the RNAs in the cell created, right? Because if we know when the RNAs in the cells are created, then we know that what transcriptional programs the cell was implementing when it created those RNAs, right? Because what it was doing is that it was creating those RNAs. So asking when an RNA was created is the same as asking how old the RNA is. And that's the fundamental insight, right? And so what we did was that we built this system that uses RNA editing in order to estimate the age of an RNA. So RNAs start out and they have 4 bases, as many of you probably are aware. And then there are enzymes that change adenosine, which is one of the bases of RNA, into inosine, which is another base, which is a non-canonical base of RNA, which gets read out by all of the transcriptional and translational machinery as a guanosine. So they're basically A to G edits, right? And what we realize is that as RNAs get older, they accumulate more A to G edits. And so by counting the number of A to G edits, you can estimate how old the RNA is, which means that you can look back in time and you can see when the RNA was made. Okay, so just fast forwarding a little bit, this is an example of what this actually looks like. So if you have RNAs in D down here, if you have RNAs that are 1 hour old, you see there's there are some edits, but not very many. 4 hours old, there are more edits. 8 hours, there are even more edits. 12 hours, there are even more. It just it accumulates over time. And then you can make this fun graph where for a particular RNA, you can say, based on the number of edits, what is the likelihood that RNA was created at a particular, during one particular hour, at a particular point in time. And you can localize the age of that RNA to, about a 3-hour window with 95% confidence. So this is pretty cool. And then of course, the actual thing is that when you actually have a cell, the cell creates more than one RNA at a time, right? It creates many RNAs in a particular transcriptional burst, which means that you can go through and you can, let me just see what, this is useful. So if you go and you look in a cell at the sum totality of all the RNAs, so you take a particular kind of RNA and you look at how many edits there are on all of those RNAs, and you make a histogram of the number of edits per RNA, right? You can imagine that you get some kind of a transcript of a graph that might look like this, what's here at the top, right? Where you have a peak of RNAs corresponding to, or a peak corresponding to newer RNAs and a peak corresponding to older RNAs, right? Because as time goes on, this graph moves to the right as the RNAs get edited more. Right. And so this is just an experiment where we induce 2 different transcriptional bursts and show that you actually get out 2 different peaks of 2 different peaks corresponding to these 2 RNA populations. You can build an algorithm and you can actually back out from this kind of data what the underlying transcriptional programs are looking backwards in time. Okay, that's the story. I don't want to bore you guys too much with the details, or you can read the paper or get in touch with whatever. The thing about this that's instructive that I thought you guys would be interested in is that, is actually to tell you a little bit about how this came about. So this whole idea started in, 2016, early 2016. Paper comes out in 2020. Oh, we're going to look backwards in time. This is the timestamp thing, right? So we're going to look backwards in time. So 2020 is when the paper gets published. In 2016, we started working on it, and we started working on this idea of temporal dynamics in RNA, but it had nothing to do with this. It was entirely trying to figure out how can we record neural activity into sequencing. How do we infer from sequencing data when neurons were active? And so in order to do that, we wanted to be able to figure out you know, when the neurons express calcium, for example, when the neurons were full of calcium, because that's a signal that, you know, the neurons are firing. And we thought that actually like an easier problem would be to measure when the neurons express CFOS, because CFOS is a marker, like when neurons fire, they express CFOS. Okay, and so we were thinking, okay, how do we tell when CFOS gets expressed? Oh, I'm telling this story a little bit wrong. We'll get back to that. So we were first on calcium. And so here's what we thought. We thought we could have these two enzymes, one of which adds adenosines to the 3' side of an RNA. So you have an RNA and it's getting A's added onto the 3' side. And then sometimes and then you have a calcium dependent enzyme, which is adding U's to the 3' side. So you can imagine that you go and you do 3' sequencing of the RNAs and you're going to see a poly A tail. There's a bunch of A's and then some U's and then a bunch more A's and then some U's, et cetera. And that would tell you there was like a burst of calcium and then there was like no calcium and then burst and et cetera. This is complete nonsense. It doesn't work at all, right? And the reason is that when you add U's to the RNA, then the RNA gets degraded. It's like a signal for degradation. Like U's on the 3' side of an RNA is a signal for degradation. That's like only one reason why this didn't work. This would be a lot of reasons why it wouldn't work, but it's fine. So then we thought, okay, maybe an easier problem is to use CFOS. And so then we started thinking, you know, still thinking about like A's and U's. We started thinking like, how do we measure like how can we figure out when CFOS was made? Or like, how can we figure out like when the neuron is expressing CFOS, which then led to this idea of like this key insight, which is that what we need is to record the age of the RNAs, and led to this idea about using ADAR. Okay, we had that idea in November 2017. We were done with experiments. The last time we did an experiment on this project was May 2018. So between when we had the idea for the project and when we were done with experiments, it was 7 months. All the data that you are looking at was generated in 7 months between November 2017 and May 2018. We submitted it in like July, June to 2018 to Science. Science reviewed it and rejected it. We then submitted it to Nature, which declined to review it. We submitted it to Cell, which reviewed it and then rejected it. And then we did a bunch more work. We added some single cell data. And then we sent it back to Science and they responded and said, please never send us this paper again. They actually reviewed it a second time. So this is actually something that like in the UK, you guys really need to know, because like Americans publish way better than British people do. And I didn't understand why at first. And then I understood, which is that like in the US, it is widely known that if you send a paper to Science, it gets rejected, you can just send it back. Like you can just like do the revisions and send it back. And like, I think people here, like they don't, like my colleagues, they like don't know this necessarily. It's like unclear. But when people hear the paper gets rejected from Science, they just send it somewhere else. And like, no, we don't do that. You just keep going until they take it. Or like this thing that has never happened, I've never seen it happen before, which is that they actually email you and say like, please, we don't want to see this paper again. So that's fine. We then actually, and like in all cases, like the reviewer comments were that the reviewers didn't understand what we were doing. And so we rewrote the entire manuscript and basically changed the narrative. Didn't change any data, didn't add any data, didn't change anything. Rewrote the entire manuscript and changed the narrative so that it was around RNA age instead of around like this whole idea of like, because it was originally this idea of like inferring transcriptional programs. People had no idea what we were talking about. We just rewrote it so it was all about RNA age, and it sailed through Nature Biotech, no comments. Like, and I was actually kind of disappointed because if we had rewritten the paper before sending it to Science the second time, it definitely would have gotten it. But it's like, so some lessons, right? Lesson number one is that like the narrative really matters when you're writing these papers. Lesson number two, so like, you know, if the reviewers don't understand it, it might not be that they're dumb. It might be that you're doing a bad job of explaining it. So this is really important to remember. Lesson number two is this thing specifically about technology development. This is not true of science projects, but that technology development projects, they go really, really fast. Like when you find something that works, like this just worked on the first try. And that's why it only took 6 months. When you find something that works, it goes really, really fast. And so if you're trying to do technology development, it does make sense to like, you know, try something. If it doesn't work, give it up and try something different. Because when you hit on something that is going to, that like works, it will just go. And like, even, you know, even like SlideSeq was from the day that we did the first experiment to when we submitted it, it was like 18 months or something, which again, compared to like projects that you do in science are like really, like science projects, or they just take much longer. So tech dev projects are very short. And yeah, and I guess the third thing is that like if a project fails, you have like projects never really fail. You like always learn something, right? So like this A and U project that we were doing with like adding A's and U's to the 3' side didn't work, but like that was what ultimately gave rise to this project. So yeah, just be flexible in your thinking, I guess, and you know, if something isn't working, like figure out what it's trying to tell you, and then you know maybe think about other approaches to the same problem. So, okay, that's my wisdom. If anyone has questions, I'm happy to hang around for a few more minutes and chat.
I have a quick question regarding the science communication side of things. So how would you recommend we think about creating a proper narrative? Are there any tips and tricks that you really think are vital?
Yeah, like, so for me, it's just important to remember that you're pitching your science. So it's not like, you know, people, I guess you can have this idea that like what they're doing is that you just need to tell people how it works and then they will understand and then they will appreciate. That is not the case, right? Like the science communication is like an active, it's like a very active thing that you have to work on, right? This is the other reason that like the labs that are really good at publishing science papers, they don't do better science usually, right? They are just way, way better at explaining what they did and why, and explaining it in a way that makes it seem completely transformational, right? Like I could take this project and title the paper, you know, a study into the dynamics of ADAR on something, like to study into the dynamics of ADAR editing a synthetic transcript, and no one would care, right? Or I can say, look, we figure out how to look backwards in time and like infer the history of RNA of like gene expression from a single endpoint measurement, and all of a sudden it's the coolest thing ever, right? So it's just like, it's really pitching. You have to figure out what is like exciting about your work, and you have to figure out how to tell that story. So yeah.
Thank you.
Yeah, someone. I don't have the names here. So one of you two who has your hands up, you can just.
Hi, I'm Angela. Hi, nice to meet you.
Hey.
Thank you so much for your very insightful presentation on your personal experience and the new technology that you developed. I'm studying neuroscience. I'm personally really interested in connectomics, the idea of mapping out the entire brain, connectomics, like between every individual neurons. I think it's a very exciting technique. I wanted to ask, what do you think it would tell us, mapping out the entire connectomics of the brain?
This is great. I will respond with another question, which is that imagine that it is the year like 1960, and someone sends you an email. I know it doesn't make any sense. Just go with me. Someone sends you an email with a text file with 3 billion A's, C's, T's, and G's in like whatever, 23, how many chromosomes are there? 26, 23, 26. I don't know. I'm a physicist, guys, okay? Like, you know, we're just like grouped into like however many chromosomes there are, right? What would you learn? The answer is like from the list of A's, C's, T's, and G's, you're not going to learn anything at all. Like the insights that we get out of sequencing the genome, right, are like the insights that we get out of sequencing the genomes of many people and being able to associate those, like the variants in those genomes with disease, right? And so likewise, what will we get from the connectome? Well, we're going to do some bioinformatics on it, right? And like, you know, you would probably, if you just have the A's, C's, T's, and G's, you'd probably be able to infer some like structure, right? You'd probably be able to like maybe identify that they're like these open reading frames or something. you like identify the ALU repeats, right? So stuff like that, like we're going to be able to identify like some basic structural elements like that. But are you going to, but like otherwise the power of connectomics is like, you know, an individual connectome doesn't tell you anything, but the ability to do connectomics is going to be extremely powerful for like disease, right?
Thank you so much for answering. I think when we have the data, especially we now live in the big data era, I think analyzing and interpreting brain data is also very important. I personally think that I'm always curious about like this gap in neuroscience. I think there exists a gap between brain activity data and behavioral output, as in techniques measuring brain data, brain activity is either the resolution is too low for individual to map out individual neural activities for disease, establishing causal relationships between behavior and disease. But then at the same time, how do you decode brain data, say for example from fMRI? What does this pattern of brain data mean, and how do you correlate it with behavior? How do you establish causal?
So I think that for that kind of thing, like basically what you need to do, well, I think that one of the most important things is having access to like data from humans, because like then you can actually, like what you need is you need to like, the goal of neuroscience is to like understand behavior and understand cognition. And I think the problem is that like in mice, you can't ask them about their cognition. That's like fundamentally the issue. And so you're measuring all this neural activity and you can't like interpret it because you literally just don't know what the cognition, you don't know what the mice are thinking, right? And I think that that's a fundamental problem. So that was actually the idea behind Siren 5. And if people are interested, I have a TED talk on that kind of, on like those questions that you can look up. But I think I can do one more. So Turga, do you want to go?
Yeah. Thank you again for your talk. It was really, really interesting. I just wanted to ask about bioentrepreneurship. So in terms of your experience with like the US and the UK, do you think we're in like a good ecosystem for bioentrepreneurship in the UK, or like, is it better in the US?
So there is no question that it is better in the US. But are you in a good ecosystem? Yes, you are. I think that if you are entrepreneurial, you're going to do great. Like basically almost, if you're entrepreneurial in London, you're going to do great. The issue in London is that most people, people get a little bit beaten. They get like the entrepreneurship a little bit beaten out of them because there's like this cultural, it's like culturally unacceptable to be like, the ambition is like kind of culturally discouraged, right? And like you have to be extremely audacious to be an entrepreneur, because you're going out there and you're saying like, yeah, I'm going to go and do this thing. And like, I'm going to go build a company that's worth 10 billion pounds or whatever. And most British people are like not comfortable saying that. But you actually literally can, like you definitely can build a company that is worth 10 billion pounds. And and if you believe that you can, I mean, like convincing yourself that you can and then being able to convince other people that you can is the way that you do it, right? And there are plenty of resources here for the entrepreneurs that are here. And you know, it's also not hard to go to the US. Like if you find that your career is limited, it's not hard to go to the US for it. But you don't actually really need to. You just need yourself to be entrepreneurial. Like the main advice that I give, like the kind of thing that you see here, and the main advice that I give people, like you see people here, they do pitch competitions. And I tell people, this drives me nuts. I'm like, guys, like stop doing pitch competitions and just pitch. Like literally just like go to real VCs and pitch real ideas, right? And like ask them for real money as opposed to like, you know, you put the same amount of work in, but you're going for a 20,000 pound prize as opposed to just doing a 20 million pound pitch. And like, why? It's because like people are kind of uncomfortable. They like don't think that they should be asking for 20 million pounds, but like, no, go do it. Like the economy, you know, when the economy is the way it is, like go find a friend and go raise 10 million pounds just on the basis of the fact that you guys think you're going to go do something, right? Like, you know, and then you'll figure it out. So if you ever need anyone to like tell you that, you know, if you ever need any encouragement, let me know, because I will just tell you that you should go and do more audacious stuff. But yeah, I think that the limitation here is cultural. It's not resource. So yeah, it's not as good as the US, but that's not the limitation that most people face, I think.
Thank you so much.
Of course. Okay, so yeah, again, I'm gonna have to go, but feel free to get in touch. My email is on my, I think you should be able to figure out how to get in touch with me on my website, or
your email down in the chat box. So for those who weren't able to ask their questions, they can shoot
perfect. Yeah, perfect. Like I said, I have a lot of open positions in the lab. So if you're interested, like get in touch, I would love to, or also collaborations or whatever, I would love to hear from you. So
thank you so much for coming along. We really appreciate everything. Take care. Have a lovely evening.
You too. See you later.
Bye.
Bye.
And thank you everyone for joining us today. It was a very, very nice informal talk, and I hope you guys enjoyed it as much as I did and we all did. And make sure to buy your memberships, and we look forward to seeing you at our next events, which you can keep up on our social media for those updates. I will stop recording.